Good morning, and welcome to Claret Bio's quarterly webinar series, the Series Talk. Our goal for the webinar series is to provide an opportunity for our customers and collaborators to share their work with a wide range of audience members and engage in discussions amongst other academics in the field. At Claret, we specialize in the development of NGS library prep and analysis tools that empower researchers to get the best data from their most challenging samples. Our flagship product, Seriously, is a single-reaction, single-stranded DNA library prep kit known for producing complex libraries from single-stranded, double-stranded, and nicked DNA. Our newly launched RNA-seq kit, Really Are None, is equipped with a downstream ribodepletion method designed to generate truly directional libraries without the need for second-strand synthesis. If you'd like more information on our products, check out our website or contact us at info@claretbio.com. As we progress through today's presentation, please submit your questions via the Q&A box at the bottom of your screen. Also, if it's not working, feel free to submit your questions into the chat box, and we will dedicate the last 10 minutes of today's webinar to addressing your queries. Now, without further ado, I'm delighted to introduce our guest speaker, Mr. Brent Lehman. Brent, a Wisconsin native, earned his undergraduate degree in biochemistry and molecular biology from the University of Wisconsin-Eau Claire. He continued his academic journey at Iowa State University, obtaining a master's degree in biochemistry. In 2016, he relocated to Madison, where he serves as the lead cell line engineer at the Advanced Genomics Editing Laboratory at the University of Wisconsin-Madison. Notably, in 2019, he extended his role to include the position of lead ancient DNA biologist at the Biotechnology Center. Since then, he's been actively involved in the MIA-related projects and is eager to share some insights with us today. We're glad to have you here today, Brent, and we're looking forward to your presentation on how modern methods are overcoming ancient problems in the missing in action recovery and identification project. Take it away, Brent.
Emily, thank you very much for the kind introduction. And so my name is Brent Lehman. I'm here today to talk about the work that the Missing in Action Recovery and Identification Project at the University of Wisconsin-Madison has done with an emphasis on our identification workflow. First, before we begin, I do want to, as Veterans Day was this past weekend, I do want to recognize the sacrifice and commitment that our current and former veterans have had to this country and for keeping us citizens safe and able to enjoy our freedoms. Here are my disclaimers. In short, I encourage you to read it. In short, I'm not endorsing anything, nor am I advocating for any product. And all of this is a research-based work done in a research setting. The University of Wisconsin Missing in Action Recovery and Identification Project honors a sacrifice of brave Americans who paid the ultimate price for our freedoms. The primary mission is to recover missing U.S. military personnel from past global conflicts. Additionally, the initiative educates students and communities year-round on the importance of past national service and help bring closure to the families of the missing. The goal of these actions is to educate students and communities throughout the year and highlight the importance of past and current national service. Many undergraduate students and volunteers aid in these core aspects. UW-Madison is the first academic institution to partner with the DPAA for repatriation, and that happened in 2015, 2016. And together, we work to identify the 82,000 U.S. service members who are missing in action over the past 80 years. Our workflow pipeline is to first survey the readily available information that can be obtained in-house, which falls into three general categories. First is a data survey, historical analyses, aquatic versus terrestrial landscape, and obstacles and potential shortcomings. All these are evaluated. Once the team is convinced and we have the resources for the mission, we then begin the recovery step. This involves two aspects of two main missions: a scout mission, and then finally a full excavation. Once complete, elements that are retrieved are handed off for sequencing. First, elements are extracted for genomic material. The material is captured in a library and either sequenced or further enriched for genomic material. Data that we do gather during these steps is turned over to the DPAA for final decision making, including identification. The sequencing aspect is what I'll be focusing on, but I wanted to first give an overview of the technology and work that is required to even get at the sequencing point, as it truly is an interdisciplinary collaborative effort. As it happens, UW-Madison is rich in human resources to accomplish these recovery project missions. In terms of project workload, the UW has faculty, student volunteers who are able to perform historical analyses of airplane crash sites and trajectories, as well as perform archaeological and paleontological analyses. We employ this group to examine the current day X files, which is a type of documentation of each unknown. Ideally, these files contain a record of any potential remains and what has been done to identify them, but that's not often the case. This team also compiles witness statements, identifying media such as dog tags or wallets, written documentation like diaries and newspapers associated with the known incident. And this is all done to formulate an answer to the question, is this a retrievable, or can this be a feasible retrieval mission? A limitation in recovery missions today is a understandable one. It's terrestrial versus aquatic. Most recovery missions to date have been of the terrestrial variety for obvious reasons. However, at least half of the 82,000 missing are at sea and are presumed to be water losses. Retrieval of these remains difficult, especially at depth, but the technology is improving. In the Southern Pacific, drones have been used to identify ship and aircraft remains. And this work has primarily been done by Andrew Petruska and his team at Scripps. Our in-house attempt at overcoming this limitation is to use eDNA to evaluate genomic material of the area. In the lower right, there's a diver in Saipan who is collecting samples that was taken back to our lab for processing. Focusing on the terrestrial cases, many of these cases are indeed very cold cases with minimal leads. Further hindrances, eyewitnesses are lost every year. Information is lost in private collections. Human identifiable elements preserved in the environment are decaying. And infrastructure development in Central Europe has made retrieval pretty difficult. The common theme of all these is that it's essentially a race against time to retrieve what we can. Additionally, there are political restraints in terms of governments in the Asia Pacific that are not collaborative to our efforts. And we certainly respect the sovereign authority of those nations to our efforts. When a site is determined that there's enough complementary information regarding its veracity, a scouting mission is performed. So scouting missions are an opportunity for our staff to connect to the local community of a potential excavation site. Oftentimes, firsthand or secondhand accounts and information is uncovered during these missions. Scout missions are also a great time to sample sediment cores for sedimentary DNA analyses, which I'll talk about later. So what I've shown on screen is in October of this past year in 2022, was a survey of a potential site with graduate students and staff. During the scout, geographical evidence of a large crater was uncovered, and you can see that on the left. This can be due to a crash or potentially a bombing. At this site, there are also key eyewitnesses and their accounts of temporary grave sites that are of known and modern location. So this scouting mission was done in collaboration with a family member who was able to provide some historical context and provide local connections to us. And so many of our initiatives do begin with family members who are committed to finding out what happened to their loved ones. We don't reach out to families. We don't want to bring up a painful subject, and we don't want to provide false hope. This is a research-driven initiative. If we know a family member is looking for information, we can be a resource for them. but we don't seek them out. Once the scout mission is complete and we think we have enough evidence to initiate an excavation, one is performed. Here's some work that was performed on recent missions. This work is best described as hot, wet, and really, really dirty. I do have a little back and forth with the field team members about who works in more miserable conditions. Their work may be hot and wet, but at least they get to see the sun. In the clean room, it's not so lucky. On-site location digs take about a week of time, and sediment is screened using sifters, as you can see in the upper left. Oftentimes, pieces of metal and debris are found using this method. In the upper right is a team looking at which areas of the sediment to screen next. At the bottom is a panorama of the scope of the excavation site, just so you can get an idea of the size of the operation in the field. In this particular case at the bottom, the site is in a cornfield, and this particular farmer was actually a direct witness to the crash site and helped inform the team where the ideal places to dig were. Most people whose property that we wish to examine are extremely accommodating and helpful, and sometimes they help excavate themselves. And we're extremely thankful to them for the property access and to help us better understand the history of the area. Once elements in the field are taken back to the lab, we do perform a genomic material extraction. These extractions and ensuing library assemblies are performed in a clean room. Our clean room is closed off with 2 individual lab rooms, one for DNA extraction and one for library assembly. A little more about the room is it's a positive pressure airflow room, so air flows from the library assembly room through the DNA extraction room and then out to the assembly area. It's extremely important that this work does get performed in a clean room. Introduction of outside material can contaminate ongoing projects and the material itself. Working in the clean room and any compromising of the integrity of the sample itself is detrimental to our goals. When I'm handling bone powder, if the elements happen to be bone, when I'm handling bone powder, I typically work with anywhere between 60 and 250 milligrams of material. When I'm working with soil, I generally need a little more than that, 200 to 500 milligrams of material. These extractions require constant agitation, whether they be sediment or other material. Again, extraction is source dependent, and we are mindful of extractions that may introduce inhibitors during our purification process. The standard in the field for materials like bone powder is pretty well established. It's just a minimal buffer with proteinase K with constant agitation. And so in our in-house samples, they're slowly rotated using a microfuge rotator. Cleanup of the bone powder is done using a silica-based column that's commonly used for PCR-based cleanups. When extracting sedimentary DNA or environmental DNA, it's important to consider the balance between extractable DNA and inhibitors that may be co-extracted. Using the same protocol for sedimentary ancient DNA as bone powder, that actually co-extracts a large amount of inhibitors, at least according to this publication. However, precipitating out the inhibitors may also incidentally co-precipitate out our target DNA. I should note that because of this information, the Kaijin Power Soil Kit they've updated the kit to prevent excess co-precipitation of the DNA. So though now a little outdated, I'm always mindful of this figure when I perform extractions and potential issues that may arise due to this critical step, because it's easy to gloss over the extraction point. The goal of these extractions is to extract all genomic material that is present in the element. So due to exposure, damage, time, or any number of environmental factors, we expect the human DNA to be damaged, or our target DNA to be damaged. This damage can be readily seen in the length of the captured fragment, and will be much shorter compared to a modern DNA extraction. So a common saying around here is data can only be as good as the QC that's performed. What do these extracts actually look like? When we run these extracts in a tape station, these are bone powder samples, and on the left is just a subset of the bone elements that I've extracted, and you can see that visualized on the chromatograms on the right of a few select samples. And how to read this as the upper and lower detection limits of the instrument are visualized by the upper and lower peaks at the end. And everything between those is the DNA makeup of the extract itself, with the size being defined on the ladder on the left. Most extracts contain an amount of shorter fragmented DNA it would sort of expect given the conditions of the sample. However, most contain this rather large, larger fragments, which is sort of an interesting observation. We'll keep that in mind when we get to the sequencing, but sort of spoiler alert, this is all bacteria and not necessarily our target capture. Once we have the actual extracts, we actually perform a library assembly. We can go one of two ways with this. The very common approach, especially with modern DNA, is to perform a double-stranded DNA library. These involve just trimming the ends and attaching attaching the ILTS ends to the termini of the fragments. This has a little less, it's not, ancient DNA isn't quite as neat as modern DNA. And so these sort of double-stranded approaches aren't ideal. They can't really, they're not the best for assembling ancient DNA. So we switched to a single-stranded DNA library approach. This is just because the ancient DNA is highly fragmented and trace amounts in the sample. So we employed the Max Planck methodology on the left, and briefly, silica column purified DNA was dephosphorylated and melted and ligated to a biotinylated adapter with a 6N overhang of a splinter oligo, which is used as bait to pull down our captured sequence. And then from there, it's captured, or it's the captured fragments and copied using a uracil tolerant polymerase, using an extension primer containing the TrueSeq Read 2. TrueSeq Read 1 fragment is then ligated on at the end, containing a 3' dideoxycytosine to prevent annealing to the initially captured single-stranded DNA fragment. These fragments are then eluted using heat and then indexed using traditional PCR with UDI primers. The Searsley approach on the right utilizes single-stranded enhancers to stabilize the denatured DNA, and NGS adapters are then ligated on using dual splinters. So the advantage of the protocol on the left is the ability to go back and reassemble and or redo previously captured fragments. However, it's speaking from experience, it's extremely labor intensive and involves many steps and washes. We initially just used this method on the left to actually show that using in-house methods, that elements that we're extracting DNA from actually do contain genuine human DNA from the past. What we found is the key advantage of this Searsley kit was the convenience minimal sample handling time, and there's a similar science at play versus the in-house method on the left. For the scarcity, I've used both the UDI Pico Plus and Nano Plus with uracil tolerant polymerases, using the protocol without enzymatic shearing modules, and clarified beads with the small fragment protocol. So once we assemble the libraries, during the library assembly step, I introduce my bias into the library assembly using a twofold approach. I limit the extension step during library amplification, and I also use bead ratios and addition of isopropanol to bias the smaller fragment retention. As a whole, as you can see on the right, the whole library fragment size is in what we call the sweet spot for next generation sequencing on our 2x150 platforms, which is less than 400 base pairs. And do recall that these sequences on the right do contain the adapter sequences, so our actual captured fragments are less in size. So there is some sample variation, as you can see in the chromatogram, but for most part, they all have the characteristic bump around exactly where we wanted. So these libraries are sequenced on an Illumina NovaSeq X Plus or a NovaSeq 6000 at the DNA sequencing core facility at the University of Wisconsin-Madison. And sequencing analysis, when you actually put it on the run, what do you get back? Well, that's a whole lot of bacteria. It's 97% bacteria. Anywhere between 0.2 and 0.8 of reads can actually be mapped to the human genome, which is a little, it's a lot of work for 0.2% of reads. So what does it mean in terms of sequence that less than 1% of all reads actually do map to the human genome? Well, anywhere between 85,000 to 350,000 of these reads are best visualized using box plots. So we actually get only 85 to 350,000 reads back from we put in, which is not the best, but it's what we have. So the best way to visualize this is to use what I call these box plots on the right, and here you can see 4 samples. These plots show reads that are filtered and aligned in the human genome, and we removed and filtered out the duplicative reads. So all these reads are unique, and they map to the human genome. It may be a little tough to see, but some of these bars are blue and red. It's color coded to abundance, with red being the most abundant. So for a good sample, like in the upper left right here, in the purple, there are 11,000 unique reads, and about 2,500 of which are over 125 base pairs, which is extremely very good for potentially identifying the elements. However, there's such a large variation between library and library, and that's just mostly due to the source material where it originated from, and you can sort of see that variation here. So if we follow our libraries through to this point, the purple samples, clearly the best in terms of total unique reads, while the yellow and green do contain some, but not quite as much as the purple. And finally, I included a library outlined in black, and that contains minimal informative reads. And that was prepped alongside of these because this is also an important QC checkpoint for us. If any modern DNA was inadvertently included or contaminated throughout the extraction and assembly pipeline, we would certainly see it here, and just because it's less fragmented. And the box plot would show a large spike in unique longer reads. So it's at this point in the process where having a good blank is important for data interpretation. The final piece of sequencing QC that we perform is to examine the termini of the captured DNA fragments. Environmentally exposed DNA undergoes damage in the form of cytosine deanimation into uracil. In terms of sequencing, this has the effect of the C to T substitution. On the 3' side, it's just the inverse. It's G to A. We can directly observe the ancientness of our library by looking for these substitutions. In the bottom box, I showed what a modern DNA substitution rate looks like. There are minimal obvious substitution rates towards the termini. However, in the top 2 libraries, there's direct and obvious evidence of these substitutions. So taking the previous slide's information with respect to the lack of modern DNA in terms of unique sequencing reads and length, and combining it with this substitution data that we can observe at the termini, we can infer that we are actually dealing with genuine genomic information from the source material. In order to get a higher throughput of relevant read, so in order to get a higher throughput of reads, we attempt to capture informative reads using hybridization capture. And using this approach allows us to pull out the identifiable human SNPs from the library that is 97% bacteria. So first, we mix the library, adapter blockers, and other hybridization reagents. The libraries are then denatured in step 2 up here. And then we introduce the bait, and we allow them to hybridize to our library overnight. Baits are then removed with a magnet, and most non-target DNA is washed away. For our workflow pipeline, this enriched sample is then re-enriched again for just to be we find the 2x enrichment is ideal. Here on the screen, you can see some of the SNPs that we're looking for. This is from the forensic capture enrichment B2 from Arbor Biosciences. So I'm not privy to the exact sequence of these of the SNPs, but there's just a wide variety of them. It's the purpose of just sharing that information. So what does our data look like after capture? Once again, our data can only ever be as good as the QC. And here I'm showing 4 single-stranded libraries that were assembled using Seriously on the left, and those same libraries that were then enriched using capture. Once again, these are just box plots that show the unique non-duplicative reads in a given library. Just the heads up, the axes are, I might have missed this before, the axes are different. So just keep an eye out for the axis change. So in the upper left library, we see a right shift from the library to the enriched library. That's encouraging to see because the shorter fragments are much more difficult to align. So the sequencing output is much greater. So we have just a slight shift to the right. There's also a higher percentage of more than 150 base pair reads, which is extremely encouraging and shows that our depth of coverage is much greater after capture. In the lower left, here's a good library as seen by the high amount of more than 150 base pair reads. After capture, that number remains high and shows that the integrity of our library didn't change much, which is good. And there's also there is a slight right shift. You can see look at that 30. That number does decrease in the enriched library. In the upper right is a typical library with a high amount of shorter reads that slopes down as the fragment length increases, with a slight bump at more than 150. After enrichment, that proportion of low fragment length decreases, and more reads we can observe in the longer fragment length. Again, encouraging. And then finally, in the lower right is a library that did not give informative read information as just a library, and after enrichment, it still didn't give informative reads as a library. Again, negative results, we can always flip that to a positive because there are minimal long reads, and that suggests that modern DNA contamination did not make it into the workflow, and this gives more confidence in our data. So unfortunately, this is where my expertise ends. Recall, this is an interdisciplinary effort. For modern DNA, if you're trying to sequence a genome, you typically see about 20 to 30x genome coverage. For ancient DNA, that's a very high bar to achieve. So there are bioinformatic programs that can impute SNPs. By that I mean, like, because there's a G at this SNP, it's more likely there's an A at this SNP in the genome. So that's mostly how ancient DNA genomes are assembled. Good ancient DNA genomes are typically 1x covered, and using the imputation models. So that's one example of what the glimpse 2 is that's a solely human imputation model. So good genomes are 1x covered. If you recall the samples that I've been showing you along for the ride, the purple one, it was actually very close to that threshold. So we got pretty close to at least a 1x coverage. So there's a piece of data that I have not shared with you, and that's all these bones, these bone elements, they actually were retrieved from salt water. That's an extremely harsh condition for maintaining genomic integrity. So this work really does open up the work for retrieval and potential identification in the aquatic theater of MIA recovery. And I'm going to go a little quicker now. We can also do the exact same thing with sediment. Sediments are assembled in a single-stranded library approach, and their profiles look a lot different than those in the bones, mostly because the bones are as a direct source. So once again, we're mostly looking for fragments around this length, and we can see that all our extracts do have that particular bump. Assemble libraries using the exact same method. Again, single-stranded for a seriously approach. And again, here we see a very characteristic NGS sweet spot. Again, this is very encouraging that we're even able to assemble libraries, because there are a lot of inhibitors in sediment, and our extraction methods have proven out to be very good. So this is the exact same QC that was performed as the bones. Again, we're not looking for unique, non-duplicative human reads. But there's a lot less, and that's just to be expected, right? These are environmental samples, not directly sourced. But what we can do is map this information to where we know where the soil cores were taken from. And so in the upper left here is a layout of the scout mission map. And the lower left is the unique read count of these cores. And you can overlay these maps, and you can figure there are ideal places of where to dig. So in the samples that gave high unique amounts of human reads, that was the site W right here. The one that didn't give as much, but still enough, that was site I. Those that didn't have any or had minimal informative reads, that was actually P down here. So these are, combining this. So this information can be used as areas to focus on if and when there is an excavation mission. So the next steps in this would be to, at least for my approach for sequencing, is to leverage the DNA sequencing depth with the advantages of single-stranded DNA library preparation. Utilize the DNA evidence in pilot survey projects, especially using the sedimentary DNA, and expand source material to include the aquatic elements of environmental DNA. And for that, that is it. So a big thank you to all our volunteers and undergraduates. In the upper middle right here is a Belgium dig that we just got done in just this past summer. On the upper right are our undergrads who are excellent volunteers. The Lower left, the U.S. ambassador to Luxembourg actually visited us, not us, visited the team on site. And the lower right is the final dig in the Belgium. So with that, I'm more than happy to take any questions.
I have a question regarding the aquatic samples. What does that look like for your team in terms of figuring out a workflow for the extraction process, and kind of assessing good and bad samples, given?
A lot of, I would say majority of the samples are difficult to even get to that 1x coverage point. There are, even in the I showed some, I guess, more typical samples. Those can actually be those can actually use as for identification, provided I'm not assembling the library, or sorry, I'm not assembling the genome from scratch. If there is a reference, say a familial member, a family member, or a set of familial family members that did provide a reference genome to us, or reference sample, it's much easier to compare a typical sample to that than it is to assemble assemble the genome from scratch. For some of these, I can for the purple sample, I can assemble that has the high potential that you can assemble that from scratch. Other ones are, it's just the nature of the recovery process. It's just difficult.
Let's see. I have a question from Monica, who says, how big is the aquatic sampling area related to the suspected source? If you think remains are located in one location, how far does the aquatic sampling go?
Yeah, that's a good one. We're in terms of the aquatic samples, we're mainly limited to the direct evidence of where that site occurred, or where the event happened. You kind of hope that there are at least elements that are immediately retrievable in that immediate vicinity. It can be difficult past that, though. That's sort of why we're looking at at least the environmental DNA, the sampling around the sites, just so we can if there's a chance of a potential location nearby the crash site, that there is human DNA that can be identified there, and is that could that be a target location that we can look at and examine and potentially excavate. So yeah, it's difficult. It's tough. We're mostly restricted to the immediate crash site, but we are trying to find new ways to search beyond that immediate crash site.
And a follow-up question from her was, what volume of seawater is retrieved for DNA extraction?
For seawater, I try to get at least 3 liters. It can be difficult to pass that onto a filter, but slow and steady centrifugation, I guess, is and for filtering is the best way to do that. And seawater can be tough, just because even in my experience, with dive teams, it's those sort of samples are pretty I'll say divers aren't necessarily molecular biologists, and sometimes it's very easy to contaminate that just from the extraction point. It's pretty easy to contaminate that sample. So of the genuine reads that we have, I try to get to at least 3 liters, but yeah, that's it's generally doable, but yeah, it's pretty, what am I trying to say? It's certainly an area that can be improved on. I think that'd be a good way to describe it.
And then I have another question from Tobin. Is there an example you can talk about where a particularly challenging sample was able to be matched to an individual? To say in another way, how degraded can these samples get while still being identifiable?
So unfortunately, I can't give specifics, but I can say that it does make identification a lot easier if there's a reference genome that we can look at. So that usually comes in the form of a family member, and it's easier. So even though I don't have like the best quality DNA, the having a narrow family member reference library to look at really does narrow the passage. And one thing we do share, like I said, we do share all our data with the DPA, and they're the ones that make the identification calls. But we have the DPA either confirm or one thing we don't want to do is provide false hope that retrieved elements are thought to belong to a family, but actually are not. And that has happened, and we do our best to try to like play both sides, if that makes sense.
And then we have another question from Chris. Are there any certain types of bones that are more ideal for endogenous DNA extraction, or are they all equally bad? Like, are teeth better? Maybe the enamel is better at keeping bacteria out.
Yes, there are. I've actually gotten some surprising good results with flat bones that you wouldn't normally consider to be good sources for genomic DNA. But in general, yes, we do tend to find that long bones are best. Those would actually be tibia, femur. I've gotten even humerus and ulna. I've gotten some pretty good reads from. There are the best recent evidence suggests the petrous bone, the inner ear bone, actually can actually hold authentic DNA far better, for like really well. So if there is a petrous, I usually try to go for that first. Otherwise, a long bone, and like you said, a tooth is generally a good, as long as I can get in from the root. That's usually a good indication that I can have a successful DNA extraction from that element.
Please join me in thanking Brent for this presentation. It was very insightful. With that, we'll end this presentation, and please feel free to reach out. We will be providing the recordings to all attendees, along with contact information should you have any other questions, either for Clara or for Brent. So thank you.